Hi guys. Having discussed the MRCS, I hope you remember what is MRCS. We're talking about the gram poor or gram staining which are not picking up the stain. So we've done the S for spirochetes already. Now we're going to pick up the next two, that is Rickettsia and Chlamydia. And in the last part of the course, we'll be doing on bacteriology, we'll be doing mycoplasma. So let's begin. Rickettsia, Chlamydia. I'm starting with Chlamydia first, which I call as ABC organism. What is ABC? There are three A's actually that you have to know out here. These are actually said to be ATP parasites. What are, firstly, that doesn't mean that, this is a parasite. No, this is very well a bacteria, but parasite is something which is dependent on someone else for something. Like a very common thing in Indian households is that your parents believe that all your friends are parasites and they're on with you for some benefit of theirs. So similarly, chlamydia is like a parasite for that matter. It is said to be an ATP parasite. That is the first thing. It takes ATP from the host. Apart from that, I think you all know that it is an atypical bacteria because it's not taking up the gram stain. And also it's very important to know that it does not grow on any of those blood agar, chocolate agar. It is not going to grow on any artificial media. Why? Because they are obligate intracellular organisms. They always need some cells to grow. They will only grow inside a cell, like a cell line or an animal. They will never ever grow on artificial media. So what are the three A's? A for do not grow on artificial media, A for ATP parasite, A for atypical bacteria. They show you blue color inclusions. That means B for basophilic inclusions are noted. Okay, now coming to the C, please note that they, as I said, cannot be cultured. You cannot be growing them on any blood agar or chocolate agar. And please remember, there is no peptidoglycan cell wall. That is why you called it an atypical bacteria, because the usual, and now if there is no peptidoglycan cell wall, now I can correlate. That is the reason that it's not picking up the gram stain, because it doesn't have the peptidoglycan cell wall. So repeating once again, what is ABC? Firstly, it's an atypical bacteria because it doesn't have the peptidoglycan cell wall. It is an ATP parasite. It cannot be grown in artificial media, or you can say it cannot be cultured, and it is going to have basophilic inclusion. So now what are the types of chlamydia that we have? There is something known as chlamydia, which has the main one, Chlamydia trachomatis, and then there is a family called chlamydophila, which is going to have the others like Chlamydia pneumoniae and Sitiki. These are the other ones that we have. So let's get going. Before I go into their, individual strains, I want to tell you that for every chlamydia, there are going to be two forms, or you can say bodies. One is known as the elementary body and one is the reticulate body. So EBRB is how it is mentioned. Elementary body, reticulate body. First, I want you to learn the mnemonic. Then I'm going to show you a big flowchart, but it's very easy if you know the mnemonic. Look at this. E for elementary body. E for, it is always going to be something related to extracellular, or in other words, I have learnt it as, it is the entry and exit wala form. Whenever you have to enter into a cell or whenever you have to leave a cell, entry and exit will be done by the elementary body. Second E is going to be equal. The DNA and RNA content is going to be equal, and E, say you will also think of all the I's. So this is going to be metabolically inactive. This is the one that is going to be anything that is entering and exiting a cell is of course the infectious form. So remember, elementary body has equal DNA RNA. It is the entry and exit wala form. It is infectious form, and it is inactive wala form. That means that when you're talking about a reticulate body, this is the one which is opposite. This is going to be intracellular. R for R, it is going to have more of RNA. R for R, this is going to be not just infectious. This is the replicating. This is the one that's going to divide. And of course, if it is dividing, this means it's metabolically active. On that note, let's start. So tell me, for example, if this is a host cell, like the human body, which is the form that is going to enter and exit? So starting the entry and exit will of course be of the elementary body. Elementary body is going to enter, but after entering, we realize that you wanted to replicate. Who will replicate? Reticulate body will replicate. So you will have to convert elementary body into reticulate body. The R for R, the reticulate body will replicate. Once it has made too many reticulate bodies, now it says I want to exit, but exit nahi ho sakta. For exit, it will again have to convert the reticulate body into the elementary body, and only the elementary body can then exit over here. So now life becomes very simple. That's simple. When you have to enter and when you have to exit, you have to take care of the elementary body. Only for the replication ability, I had got the Reticulate body. So now telling you again, for the entry and the exit or extracellular, which has equal amount of DNA, RNA, it is the infectious and the inactive form. Baki sab kuch opposite becomes reticulate. So I can say the reticulate body is the replicative form. It is the gym going wala, the bodybuilder kind of a person who is doing all the work, who is taking care of metabolism, who is replicating. That is why the size is more. Gym mein jaoge to body banega, right? So that is why the reticulate body is actively dividing, is actively multiplying, it is actively metabolically active. That is why it has a bigger size. Now let's go on to the different strains. Now I told you there's Chlamydia trachomatis. Chlamydia trachomatis only has many strains. Apart from that, the Chlamydophila, that is pneumonia and Psittaci, will be others. Let's start. Chlamydia trachomatis is going to go as A, B, C, D and so on. So first I've taken A, B and C. What does Chlamydia trachomatis A, B, B, A and C cause? It causes trachoma. I hope you remember trachoma is the most common infectious cause of blindness in our country. Infectious cause of blindness in our country is going to be trachoma, and in that also the most common one is caused by what? The most common is caused by this type B. Chlamydia trachomatis type B causes the most common trachoma. Okay, after ABC is one category causing trachoma. Now let's go on with the D's. D, E, F, G, H, I and K. Remember, these are going to very commonly cause NGU, non-gonococcal urethritis, the most common cause of non-gonococcal urethritis. So what is non-gonococcal urethritis going to present with? It's going to present with a discharge. It could be a urethral discharge in males, it could be a cervical discharge in females. So that is a discharge. And just imagine if there is, because discharge obviously can come in contact with hands, and if with that discharge someone ends up inoculating it into his or her eyes, it is going to result in inclusion conjunctivitis. So I realized that Chlamydia trachomatis ABC were also causing an eye problem, and the D onwards are also causing an eye problem. But the difference is that trachoma is something which can cause blindness, and inclusion conjunctivitis does not cause blindness. That is something which you should know as a difference. What else? So now if it is in the genital tract, this means if a child is passing through that genital tract, the child can also get it. This can definitely result in ophthalmia neonatorum, infant pneumonia. Yes, so these are the other childhood related problems it can cause when the child is passing through the genital tract, mother to child. Then you have done all the way till K. Now you're left with L's. L1, L2, L3, it causes a disease with L called lymphogranuloma venereum. L1, L2, L3 cause LGV, lymphogranuloma venereum, in which you will know most common is L2. So I have realized always the two things are most common. If I say amongst A, B, B, A and C, which is the most common for trachoma, the B one is common. Out of L1, L2, L3, most common for LGV is L2. So these were all Chlamydia trachomatis. Now we come to Chlamydia pneumoniae. So remember, pneumoniae has only one strain. It has only one strain, and that is known as the Tuar strain. Tuar for Taiwan. Taiwan mein discover hua tha. So that is the Tuar strain. Whereas Psittaci has many strains, which I'm going to study these in detail also. So note which of, so I realized trachomatis also has a lot of strains. Psittaci also has a lot of strains. Which is the one which has only one strain? So Pneumono has only one strain, rest all of them have multiple strains. Now we're going to go into detail. See, trachoma, NGU, inclusion, all this I've told you. I didn't tell you anything about lymphogranuloma venereum, so why don't I start with that? LGV, which Chlamydia trachomatis has caused it? L1, L2, L3, and that is known as ABCD, E, F, G. So note, most of the cases are asymptomatic. Pehle to many cases go asymptomatic. If there is a symptom, it is very important that they are going to have something called buboes. So primarily, if I ask you, lymphogranuloma venereum, it is going to cause a genital ulcer also. It is going to cause a lymph node. So genital ulcer is going to be painless. But since lymphogranuloma venereum having all the L's, so lymph node ko importance do. It is the pain that is happening in the lymph node, and that is known as the buboes. The painful lymph nodes are known as buboes. In fact, I'll tell you that you know which lymph nodes. Ab genital ulcer hai to which lymph nodes? Inguinal lymph nodes. Now have a look at this picture. You can see a set of, and they are very red. They are inflamed inguinal lymph nodes, inflamed buboes. Why inflamed? Because they are definitely showing you signs of inflammation. There's a lot of pain. So you can see a set of inflamed buboes over here, and you can see some lymph nodes over here, and can you see there is a line or a depression in between? I call it a groove sign. That groove is the passing of the inguinal ligament. So remember, groove is a gaddha. Okay, groove sign क्या होता है? Groove sign is going to be a gadha. Gadha means it's a depression, so that you can learn that the inguinal ligament on one side is the lymph node, on the other side is the lymph node. So I can say G stands for the groove sign. What else? Now let's go on to back to our ABCDE wala mnemonic. So the C is going to stand for that it is caused by, of course, Chlamydia trachomatis. I know it is caused by L1, L2, L3, and it is associated with the cup syndrome. What is cup syndrome? Conjunctivitis, urethritis, polyarthritis. What is it together known as? It is known as the Ritter syndrome. Conjunctivitis, urethritis, polyarthritis. Now we come to the next, that is D. What is the drug of choice? It is D for drug of choice, D for doxycycline. Let's go on to some complications. It shows you esthesiomean. What is esthesiomean? These are strictures in the genital area, that is rectal and vulval strictures. Now let's come to some tests and some complication. It has two F's in it. One is a Fry's test. Fry's test is nothing but a skin test. Fry's test is a skin test, and another F is a complication called the Fitzhugh-Curtis syndrome, which nothing but perihepatitis. Fitzhugh-Curtis syndrome is perihepatitis. I hope you remember I have taught you Fitzhugh-Curtis syndrome somewhere earlier also. I had taught you this same syndrome of perihepatitis with Neisseria gonorrhoeae, which was also a genital infection. Now I am teaching you with lymphogranuloma venereum or Chlamydia trachomatis, whatever is written in the paper. I hope everyone's okay. So quick revision ABCD. It is asymptomatic but can show you painful buboes. Chlamydia trachomatis with the cup syndrome, that is Ritter syndrome. Drug of choice doxycycline. Esthesiomean strictures. Fry's is a skin test, and Fitzhugh-Curtis syndrome is perihepatitis. G for Gadha, that is going to be the groove sign, which is a very important picture that we have. How are you going to do the lab diagnosis? So sample, I have a whole lot of options depending on whether it is Chlamydia trachomatis ABC or D2K or L1, L2, L3. I can take anything from conjunctival to urethra to urine to blood to buboes. If it's a case of lymphogranuloma venereum, I take buboes. So there's a whole spectrum in front of me. No one's going to ask you the sample. That will depend on the patient. But what is important is the microscopy. So tell me, is it going to be gram staining of any use? Of course not. It is gram poor. So something that is gram poor, what am I going to do? I'm today going to tell you a rhyme which will go on for the rest of this chapter. Giemsa, Giminas, Castaneda, Machiavello. They sound like some Dawn movie, James Bond movie wala names. They're quite heavy names. Giemsa, Giminas, Castaneda, Machiavello. So some macho James Bond movie names. These are the 4 stains. And please note, this is a PYQ, that Lugol's iodine can come positive because Chlamydia trachomatis has glycogen. Remember, Lugol's iodine can come in the glycogen matrix. But the most important is again Giemsa, Giminas, Castaneda, Machiavello. Okay, the story is not over. Do you have any culture media? No, the first story of chlamydia was that no culture media because it is intracellular organism. So how are you going to grow it? You will need cell lines, HeLa, Hep2, McCoy. You basically need some kind of a cell. It will grow in a cell only. It won't grow in a culture media. Then for everything on this planet, guys, we do antigen antibody testing with ELISA and complement fixation. For everything in this chapter and everywhere in micro, you've done PCR, nucleic acid amplification testing, which is sensitive and specific. So that happens to be for everyone. That's an end for Chlamydia trachomatis. Now I go up to that one which has only one strain. Chlamydia pneumono. Mono means it is caused by the Tuor strain. As I told you, Tuor was what? It was found in Taiwan. So it causes the pneumonia. So of course it's going to cause pneumonia, and it caused pneumonia in Taiwan. That is why it was called as the Tuor strain. So I hope you all remember this is the 3rd most common cause of community acquired pneumonia. I hope we can also know the first most common cause is of course going to be Streptococcus pneumoniae, and the second most common cause is going to be Haemophilus influenza. So this comes under the 3rd most common cause, and that is the Chlamydia pneumoniae. But is that it? No. It is also associated with atherosclerosis, and this is something I have taught you in your pathology chapters also. Which infection can cause atherosclerosis? Do you remember what were the 3 infections that I told you? One of them was definitely chlamydia. Other than that, I have also told you cytomegalovirus and herpes virus. Even herpes can cause. So bacteria, virus, both have been implicated, but chlamydia is important. Apart from that, for second year students, you can learn these because they will help you in writing more number of pages. That is, it has also been associated with sarcoidosis and asthma. But atherosclerosis is a NEET PG question. Okay, so this was about Chlamydia pneumono because one strain, but when I come to Chlamydia psittaci, it has lots and lots of serotypes. But remember, this is something P for parrots. This is something which is seen in parrots, and from parrots it comes to human. There is no human to human thing known over here. It is not human to human, it is from parrots to human. Okay, so P for psittaci and P is going to be for parrots, and what is it going to cause when it comes to us? It causes pneumonia. So yeah, that is about psittaci, and that makes an end of the Chlamydia chapter also.